introduce yourself and your project.
Hello. I'm Pavel, and I'm going to talk about our project, which we started with Anton a couple of years ago. So it's going to be kind of a mixture of neuroscience, data science, robotics, and I'm going to define one problem that hopefully you might find interesting and show you how we approached it. Like before we start, let me introduce the guys who worked on this project. It's me and my friend Anton. He's a neuroscientist in University of Sheffield, and I'm a software developer, do like software development and a bit of data science and so on. So first I need to remind you about such object as a stem cell. So stem cell is a cell that can be transformed or differentiated into any type of cell, like of human body. So for example, you can get red blood cells, neurons, fat cells, and so on. So we are especially interested in neurons. And so how it usually happens, so like around stem cell, there are some cells, regulators who command stem cells using some different signal molecules, which cell they need to differentiate to. And the question is, can we as like scientists, engineers control what stem cells differentiate to? Like, for instance, on this picture, you can see on the left side is like a group of a culture of stem cells, and on the right side there is a bunch of neurons differentiated from this culture. And can we do this in a predictable way if we want to say get some neurons? So scientists know that there are like some of signal molecules that are used in this process, but unfortunately, like they know only some of the signal molecules, and they cannot do this in a predictable way. So usually they just apply some of the signal molecules and hope that there are going to be some percentage of some cells. For instance, you can see here some like already known practices how to get some types of neurons. And the thing is, actually, there are like many different cell types. For example, even neurons, there are subclasses of neurons. And when I described some like algorithm that we scientists or like cells regulators can use to command stem cells. What type of cells they want to. We can imagine this as a set of charts, like a set of graphs. So on the Y, you can see a concentration of some substance, and on the X, different. It's basically time. So like you take different substances and you apply them in different time and in different concentrations. So basically, we need to find those algorithms for different types of cells that we want stem cells to differentiate to. Yeah, and how do we find this algorithm? So there are some calculations, and for every possible algorithm for just for one type of cells, there can be hundreds of thousands different possible, algorithms and configurations. And usually, obviously it cannot be performed manually, and even with some big robots like that, biologists have big expensive robots, they cannot do this. Like they still cannot do this level of parallelization and search optimization algorithms. So this is still a problem. And how can we kind of approach this problem? Sorry. So we can create a lot of cheap robots that can, every lab in every university has few of them, and they work collaboratively. Like they exchange data and they try to crack this algorithm together. And we've created an open source project, which so this is a robot, a fluorescent microscope, which is designed to do different manipulations with cells, not only stem cells. So it looks like a 3D printer, in terms of XY stage. And also it has a fluorescent microscope, a set of pumps which can apply different solutions quite precisely. And also, yeah, it has an objective and so on. And it works with standard 96 Wells plates. This kind of plate. So this is kind of like a standard thing for biology. So there is 96 wells, and they have transparent kind of bottom, so you can see all the content of the wells via microscope. Yeah. So Anton designed microscope, and it's a fluorescent microscope which can work in two modes. One is just simple white light, and another one a fluorescent light. So the most kind of hocus pocus thing about this microscope is the dichroic mirror, which is in the middle. You can see it. So the blue LED emits blue light. It goes through the filter. The mirror reflects it, and it goes through the objective, and it emits. It reacts with cells, and they themselves emit green light, and this green light already passed through the dichroic mirror and is detected on the camera. And on the right side, you can see just the standard stepper motor and NEMA 17, and this allows us to move the objective up and down, and this allows us to do autofocus on the samples. There's a known issue in microscopy, because because of the physics of the objectives, they all have very shallow depths of field. So you need to be very precise when you find this distance, focus distance. So, and the microscope can make these pictures. So in the middle, there is a standard target, which is used to benchmark microscopes. And like the smallest line here is just half of a micrometer. And this is like what the robot can produce. Like, so every box on this screen is one well, and it can conduct some long experiments and take pictures at different time of different cells. So, for example, on this video, the robot applies some calcium and detects how how bright that how much more light they emit because of the this calcium solution. So there are some parts of the robot. On the top, you can see on the top right side is a just like very simple basic pump. So basically, it's a syringe and a stepper motor, but it's precise enough. And you just connect a tube, and the tube goes, from this module on top. And there are like, you can connect up to 5 pumps, up to 8 pumps to this thing. On the bottom left corner is just an XY stage, and it works like a 3D printer. So there are a couple of lead screws there, and they can move this thing quite precisely. So, and on the bottom right is a robot assembled. Yeah, some videos of how it changed a liquid from red to blue. So it can like apply different solutions and work with the wells. And like another application of this robot is that we use it for data collection, and we can make pictures of many like big data pictures of different cells or microscopic objects, and use them to train our machine learning models and neural networks, like for cell segmentation, cell classification, and so. So the architecture nowadays is quite simple. So like there is a USB connection between the camera and computer. Computer connects to the our PCB, which controls the robot. It uses a serial connection, serial port as well, and the PCB controls using Arduino controls all the stuff, all the mechanical parts. So we designed our own PCB. I mean, it was first PCB we designed. It's probably, I don't know, like awful from the point of view of some professional PCB designer. We used some just open source tools, and we ordered like a bunch of PCBs in China. You buy these links. By the way, it's all open source and stored in the same GitHub repository. And yeah, there is a list of all components and you can, I mean, if you want to, you can order the same PCB and use it. Yeah, this is how it looks like when it's printed. And these are actually the most of the components which I used to control it. Is just an Arduino Amiga, 12 drivers for the stepper motors, CNC shield, which helps us to, like manipulate the drivers in a more convenient way. One MOSFET for controlling a white LED, and one special constant current LED driver for the blue LED, because like it needs to be smart, because we want the same intensity of the blue light. And this is how it looks like assembled. Yeah, like definitely we need to come up with some kind of box for it. And yeah, regarding the code, local computer, like the code for the computer is written in Python, and all the code for the firmware is on Arduino. It's written in C++. Because yeah, there are a couple of UML diagrams. I mean, they're quite simple as well, and this is firmware, and it's, I mean, basically, every box here, every class corresponds to some component, so it's kind of straightforward. And the same for the software part, which is on the computer. So this is like simple interface, which the operator of the robot can see. So the like MVP, first prototype. Hopefully, I will be able at some point to rewrite it in some like more, I know, good-looking library. And another part of the system is a camera. So yeah, I mean, there is a library for this camera and you can control it. The only thing is that you probably need to adjust is the configuration of frames series, and the logic behind the frame buffer. Like if you want to drop some frames, or you just want to analyze every frame, or you want to do this real time, and so on. Yeah, and we implemented our algorithms of autofocus as well. So you can see that on the right side, the measure of how focused the image is. And on the left side, what the camera sees. So the robot itself adjusts the distance to the sample, and yeah, like now it's focused. This is a critical part. The robot cannot analyze images if they are blurred. Yeah, we implemented some standard algorithms like Tank, Log, Lappen. I mean, this is the autofocus basically works the same in the way that it works on your mobile phones. Just like, I mean, nothing new here, but one of the things that we implemented, yeah, so all the classical algorithms, they use just basically derivatives, Sobel measure and so on. So it's basically one convolution which run through the image. But at some point, we created a simple neural network, convolutional neural network, which performs quite well for this task. And you can implement your own like logic for this robot. The biologists call all these algorithm protocols. It's kind of like name from biology. So basically, all the protocols are just Python-based classes, and you can just subclass from this base class and implement your own perform function. And like we provide a standard library which helps you to, I don't know, move the stage left to right, some microns, or like control the pumps, control the LED, and so on. So this is basically how it looks like some simple protocol. And also you can, of course, take pictures, store them somewhere, and so on. And we did some performance benchmark protocols, performance measures. So for example, we, took a picture of the same place on the well hundreds of times, and then automatically measure what's the average distance between all the versions of this picture to be able to be sure that all our experiments are reproducible. So, and it's quite precise, and you can see on left and the right sides of this image, we basically moved our stage, took a picture, moved our stage into 0, 0 position, and positioned it in the same coordinates on and on many times. So this kind of benchmark proves that it's reproducible. You can find all the components, CAD models, components, firmware, software on our repository in GitHub. And this picture shows basically the end-to-end pipeline. So the robot performs some experiments. You can see all these images in the user interface. When you take pictures, the robot can segment all the cells and track the cells individually during the time. For example, on the left graph, it shows the response, why, like how bright every cell individually, and tracks it over the time and plots the graphs. Then it creates all this like time-lapse videos and produce, you can see the behavior of every cell individually. This is a time-lapse that was taken from one of the robots in the university. So it's just, I don't know, it's probably several hours here. So it just keeps working. So yeah, and the idea is that every biologist just has this robot on their desk and can control it remotely, or I don't know, set up some experiments for the long time during the weekend and so on. Sorry. And the big idea, the big picture is what I'm working on now is I want to create a web service which provides nodes orchestration. So like I mentioned that there are many universities and many labs in every university, and every lab has a bunch of these robots, and we want to perform one global experiment, saying that which can crack this algorithm of stem cells differentiation, and we want to store all the data in one place, and we want to decide, we want to spread the load of the algorithm between all these robots. So basically, this is what I'm working now. Yes, and in this slides deck, I'm providing all the links. And thank you for your attention. Yeah. Any questions?
Hi. Yep. I'm just totally amazed that you can detect that you've actually created a neuron just by looking at it. I mean, you don't do any kind of functional test on it that it is in fact a neuron, or not just some new thing you might have created.
Yeah, this is actually true. And the good thing about all this stuff is that they are like you can distinguish different types of different cells by just their appearance, and this is what actually helps us. So basically, what is the loss function for all this algorithm cracking is like what the percentage of stem cells which differentiated to the needed type of cells. So like to calculate this ratio, you need to be able to segment all the cells and then classify them. And yeah, this is what we are working on now as well. But yeah, it's solvable. You can detect different types of cells.
I was just wondering if is there like a commercial application or a commercial version of this? Like, are you comparing it against something that's already out there, or is this something brand new that no one's ever done?
We don't know about something commercial, and I think this is because usually it is like one huge, very expensive robot for one lab, and usually nobody produce like personal robots, cheap personal robots for cells culture and something like this.
Right. So there's very expensive devices that are out there, but
yeah,
nothing like this that sort of enable small labs to like join up and do that sort of thing. That's pretty cool.
Okay, chat question. Can the system be expanded to control incubator conditions, maybe robotic extraction to place in the microscope?
Say again.
So can the system be expanded to control incubator conditions?
Yeah.
And maybe robotic extraction to place in the microscope?
Yeah, we are kind of like now. So now we sometimes put this robot into an incubator, CO2 incubator, like all the labs, they have CO2, like big CO2 incubators. But the problem is it's not very adapted. And sometimes there is like rust and so on. So we are now working on a cheaper version of incubator, which just kind of like covers the robot itself, and it shouldn't be very hard because basically you need to a temperature sensor and humidity sensor, and keep the same temperature and concentration of CO2.
Excellent. Another question was, do you have any kind of error detection or notification if something isn't going correctly and it's been left unattended?
Yeah, got it. Not yet, but this is something we want to implement as well, because like all this orchestration is just being developed. Like
Yeah. What methods are you using to manipulate the outcome of the stem cell to make it into the version that you want it to be, or how does that work?
Sorry, what?
So the stem cell can turn into multiple different types of cell.
Yeah.
So what's influencing? How are you influencing the? How are you persuading it to turn into what you want it to?
Yeah. Let me. Yeah, you see this bunch of pipes going on like from the top. So these are actually some pumps which connected to them, and every pump contains some signal molecule, or I know, like different substances, and you just apply some of the concentrations of these substances in like different proportions to the wells and stem cells cultures. So this is how you influence them, and the microscope for basically observing what's the outcome.
So how much have you actually used this device? Have you used it for lots of experiments yet, or is it still relatively new?
So I do not use it myself nowadays because like I'm not a biologist and I'm just trained, but there are, I think, around 10 robots that are already working in the University of Sheffield. They built, I think, around 10 of them. And I know that we were contacted by a couple of universities, one from China, one from other. So the guys are assembling their versions of this robot. So it's quite interesting.
How much each robot cost? Like,
It's just a couple hundreds of dollars if you like all together.
very impressive.
Oh, hi. You mentioned at the beginning that you have a very large search space. And I see you've got a central coordinating station where you collect the results. What sort of search algorithm do you use? Is it just is it a exhaustive grid search and you just divide up the domain between these remote robots, or do you have a more sophisticated dynamic search where the results inform what the experiments should be done next?
Yeah, so this stage, like we don't do yet this stage because for this stage we need more robots, but there are already several algorithms which optimization algorithms for such problems. It's basically kind of like discrete version of gradient descent or something like this. When you start in the search space in different positions and gradually move.
Yeah. But you've got several searches going on in parallel. So it's
yeah. So the idea when we get enough robots to actually perform it, then we will be ready to run all the search. So the search part is just like, you know, the design, why we do this and what we want to achieve. So we haven't completed this search part yet.
Okay. Cheers.
Okay.
Any other questions online or in the room?
Just wondering so if you're developing a neuron, for example, have you already got an image of a neuron that you compare it against, or how do you know when you achieved a neuron cell or any sort of particular cell?
Yeah, there are like we collect our own data sets to be able like train our models, and there are like some open data sets of this as well. So like we train algorithms which can detect, you know, stem cells. And another thing, like why all this search process is complex, because actually differentiation of stem cell into group of stem cells into other types of cell, it can take weeks or like even more than months. So it's quite a long one. So we need to parallelize it significantly.
Do you adjust the concentrations of the substances? Can you sort of tweak things? If you notice a cell is going in the wrong direction, can you?
Potentially, yes, but we are not at the stage when we can, just understand by, visual that the cell is going on into some, other direction. And I wouldn't say that it's possible to correct the growth if it's already kind of like chosen the path, but probably we can in future. One of the ideas of Anton is that we can detect detect it on early stages, like we not after weeks or something, but maybe days, but we cannot tell if it's possible now.
Thank you.
Yeah.
Cool. Any last questions? I think it's really interesting, and just seems like I could probably ask you back here and do an update presentation like 10 years, you're still following work on this. Just feels like it'd go forever, It's like one of those projects that's that's where there's some more chat questions.
So one of the questions is, has it been used to analyze organoids? I have no idea what an organoid is.
Organoids is a kind of like group of cells in like 3D group of cells. Like say, some people grow brain organoids. It's like a group of neurons which connected all together. It's small one. And no, we haven't used it yet, but we did some stuff not only with stem cells but with HeLa cells. HeLa cells is like another biological, interesting biological object, which is kind of like a tumor which was extracted from one woman 10 years ago. She died, but this group of cells still grows, and every lab in the world basically has parts of her cells, and they're still growing.
There was one other question. Can the setup be modified to track morphological changes in the larval stages of flies, for example, or their responses to environment?
I'm not sure. I mean, I'm kind of like don't understand this domain. I mean, I'm not a biologist myself, but probably.
Thank you.
Thank you so much. Oh, there's
all the parts. Is this 3D printed, or it looks like there's a lot of different materials. Are you
Yeah,
3D printing parts of it, or is
yeah, all the plastic parts are 3D printed. So all the frame is like basic aluminum extrusions that everybody use for building 3D printers. So you can order them online.
Cool. Well, thank you very much. It's really very interesting.
Thank you.